So now it's my great pleasure to introduce our last speaker, Vikram Paralkar, who is a physician scientist in hematology and oncology at the University of Pennsylvania. So Vikram obtained his MD degree in Mumbai and then performed his residency as well as fellow period at UPenn. So he spent quite a lot of time there. And he actually was trained with Dr. Mitch Weiss as a postdoc, focusing on the non-coding RNAs in blood lineage. He then started his independent position at UPenn with a particular focus on rRNA regulation as well as nucleobiology, in particular how their importance in hematopoietic stem cells. And he has won many grants for this type studies for his lab. He has won a lot of awards, such as the fellow as well as faculty scholar of American Society of Hematology, as well as the NIDDK KO8 grant. So he has a lot of grants for studying his RNA regulations. And with that, I would like to welcome Dr. Vikram to present his research. I think you can start sharing your screen. Yeah.
Thank you so much to Sky and to the organizers for the opportunity to present work from my laboratory. And I really want to congratulate the entire Fragile Nucleosome group for this community that you started during the pandemic and for keeping it going strong. So really happy to be here. So as Sky mentioned, I'm a leukemia oncologist, and my laboratory studies hematopoiesis, which is the process through which stem and progenitor cells in all of our bone marrows are producing over 100 billion mature blood cells every day that we require for our health and survival. And I'm specifically interested in understanding how transcription factors and chromatin proteins occupying different regions of the genome turn on or turn off different coding as well as non-coding RNAs that collectively set the behavior of normal cells, and how mutations in these regulators or dysregulation or co-opting of these regulators occurs when you have malignant transformation. So all of you are familiar with ribosomes. These are essential molecular machines in every cell. You know that they have a large 60S and a small 40S subunit, and each of these subunits contains RNA components listed here in blue, and a total of 80 RPS and RPL proteins that collectively make up the ribosome. And as you can see from the structure over here, from these dark green and light green loops, a majority of the ribosome, both by volume as well as by mass, is actually composed of RNA. And because there are several hundred thousands to millions of ribosomes per cell, ribosomal RNA is the most abundant RNA in the cell, which is of course no surprise to anyone who's ever run RNA in a gel. Most of what you see is indeed 18S and 28S rRNA. Now, the biogenesis of these ribosomes occurs in the nucleolus. So here are some acute myeloid leukemia blast cells. This is the disorder that I specialize in treating, and they have these characteristic prominent nucleoli. But in fact, many malignant cell types have larger nucleoli compared to their normal counterparts. And what's happening within these nucleoli is the transcription of 47S pre-rRNA by ribosomal DNA by polymerase I. And it's this 47S on nascent rRNA that goes on to get processed and cleaved and packaged along with ribosomal proteins to eventually form mature ribosomes that participate in translation. Now, my lab is specifically studying this process. And just because this is often a source of confusion, what I'm not going to be talking about today is the transcription of coding genes that make mRNAs that go on to produce the proteins that become part of the ribosome. There's a lot of interesting biology there, but what I'm going to focus on is the transcription of the non-coding RNA that becomes most of the mass of the ribosome. Now, these RNAs are transcribed from these very unusual genes that are present in repeats across 5 different chromosomes in humans and mice. So each one of our diploid cells contains 10 arrays of rDNA, and each of these arrays contains these head-to-tail repeats of our near identical repeats, each of which contains a promoter, a 13 kilobase transcribed region, and then a 30 kilobase intergenic spacer. And each of these units is about 45 kilobases in length, and there are 200 to 600 of these copies in the diploid genome of each cell. And these are by far the most intensively transcribed genes in the genome. So here is a classic electron microscopy image from the 1960s. It's called a Miller spread. And the central axis over here is the DNA. And you are looking at 2 genes each one of which is loaded with a conveyor belt of about 70 to 100 polymerase 1 molecules that are processing through and radiating out these very beautiful nascent rRNA Christmas trees. So they're often called as Christmas tree spreads. And every cell in your body is transcribing several of these and transcribing about 1,000 to 10,000 rRNA transcripts per minute. So really, there are no other genes that are transcribed with such intensity. Now, in hematopoiesis, you have cells of varying sizes, functions, proliferation kinetics, and as it turns out, rRNA transcription rates. So there's about a 5 to 10-fold variation in the amount of nascent rRNA as you go from stem to progenitor cells to mature and differentiated cells. And this raises a couple of different questions. The first question is, how do different normal populations fine-tune their rRNA transcription rates? Second, can cell type specific aberrations in rRNA transcription cause cell type specific abnormalities? And third, are cell type specific rRNA regulators being co-opted to drive abundant ribosome biogenesis in cancer? And even though my lab is interested in answering these questions in the context of hematopoiesis, you can really think of these as sort of broad questions that could be asked about rRNA transcription in multicellular eukaryotes. Now, the principles of cell type specific regulation of Pol2 transcription and coding genes is actually quite well understood. We know that every cell has the same genome and has the same gene sequences, but what is different between different cell types are cell type specific transcription factors. And these occupy regions that we call promoters or cis-regulatory elements or enhancers, and collectively they modulate the core universal machinery, which is Pol2 and its co-activators, and recruit them to specific genes to drive their transcription as required for that cell type. And here on this image on the right over here are shown several different regulators that are required for either hematopoietic stem cell identity or lineage differentiation. So these are cell type specific transcription factors. And so we asked, do cell type specific transcription factors regulate Pol1 activity and rRNA transcription? And so what this would look like is there may be different transcription factors expressed in different cell types. And in each cell type, in a combinatorial manner, these factors may occupy either the promoter or different regions of these RNA repeats and fine-tune the amount of ribosome production that is occurring in each cell type. And one of the reasons I would say that this has simply not been explored systematically in the past is because ribosome biogenesis is often presumed to be a housekeeping process, and also because the bioinformatic pipelines that we use typically do not give any rDNA mapping reads. And so rDNA has pretty much become somewhat of a bioinformatic blind spot when it comes to mapping. And the work I'm going to be talking about to answer this question was performed by two really outstanding members of my lab. The first is Charles Anthony, who's a postdoctoral fellow, and Subin George, who's a computational biologist. So we wanted to approach this question with the broadest possible lens. And so the first thing we did is compile a list of transcription factors having any role in any cell type in hematopoiesis. And so we first looked at the published literature. We went to hematopoietic specific CRISPR screen dependencies. We looked at transcription factors that are mutated in hematological malignancies. And we also included all members of large transcription factor families where even a single member has been known to have a role in hematopoiesis. This gave us a list of about 192 candidate transcription factors. It's worth noting that there are about 1,000 annotated transcription factors. So we are covering about 10 to 20% of all transcription factors with this approach. To this, we added 134 chromatin complexes, including nucleosome remodelers, histone readers and modifiers, looping complexes, cohesins, etc. And then we went to ENCODE and GEO and tried to identify as many human or mouse ChIP-seq datasets that have been performed and deposited by investigators such as yourselves. And we then downloaded the raw sequencing files for those datasets and mapped them to these specialized genomes for human and mouse in which an extra rDNA reference sequence had been added on. And what we now have is a large database of about 1,200 human tracks and 900 mouse tracks for spanning about 200 to 250 factors. And specifically because we're interested in transcription factors, we have 155 human and 123 mouse transcription factors represented in our atlas. And to cut a long story short, what we found was that numerous hematopoietic and leukemic or oncogenic transcription factors bind to rDNA. So to the left are human tracks, to the right are mouse tracks. We're looking at the reference rDNA sequence. And each of these tracks is actually an average track for dozens of tracks with similar expression patterns. So we found tracks that had broad expression across the rDNA sequence. There were others that had sharp peaks at the promoter, others that had a peak within the transcribe region, and there were others that had peaks within intergenic spacers. Many of these factors had not been reported before as binding to rDNA. And so for many of them, we still don't know what they are doing binding to rDNA. In the interest of time, I'm only going to be able to talk about one of them. So this is the CBP family of transcription factors. These factors have diverse roles in normal as well as leukemic hematopoiesis. And we found 19 human and 13 mouse tracks that showed the exact same peak in human and mouse rDNA at a canonical motif sequence. Keep in mind that these are chip seeds done by different groups using different antibodies in different cells, in fact, for different proteins, because these are different members of the same family, and using different sequencing protocols. And yet they all showed the exact same peak. To our knowledge, this peak had not previously been reported on rDNA. Now, CBP factors belong to this larger superfamily of transcription factors known as leucine zipper or BZIP transcription factors. And they're called this way because they have this leucine-rich region that allows these factors to zipper together to produce an adjacent dimeric DNA binding region. And this is what determines its sequence specificity. Now, this particular family has 6 members, alpha, beta, gamma, and so on. It evolved early in holozoa, so it's not found in fungi, and it diversified through metazoan evolution to the point where there are 6 family members in vertebrates. And these CBP factors are known to bind DNA as homodimers or heterodimers, and they're known to regulate promoters and enhancers of FOL2 genes. Now, each CBP family member has its own cell type specific expression pattern, its own function, and its own knockout phenotype. We specifically were interested in CBP alpha because it's been known for 20 years now that knockout of CBP alpha in the hematopoietic system causes loss of this population known as the GMP population, or the granulocyte monocyte progenitor population. And as a result, you lose the downstream cells, which are granulocyte and monocytes. And so this is what the blood count from the mouse looks like. The red cell levels are fine, the platelets are fine, the lymphocytes are fine, but what you have is an absolute absence of neutrophils and monocytes, and these mice die within the next few weeks of life-threatening infections. And so in essence, what you have is a factor that is essential for survival, but in a very cell type specific manner. It is not required in all cell types. A CBP alpha knockout has also been shown in large CRISPR screens like DefMap to be selectively lethal in AML cell lines. And this factor has gain of factor function mutations in 10% of AML. So it was a very high yield factor for us to go after to look at rDNA regulation. And so what we did was we took a mouse cell line that is the counterpart of the endogenous population in which this is required. So this is the granulocyte monocyte progenitor cell line. And the CBP alpha gene is just 1 exon. And we used CRISPR HDR to tag the FKBP and flag domain into the C-terminus end of the CBP alpha gene. And so what we now have is a fusion protein where when we treat the cells with DTAG, it recruits an E3 ubiquitin ligase and degrades the fusion protein. And what this looks like is here's the parental cell with its isoforms of CBP alpha. Each of these bands is shifted 15 kilodaltons up because of the addition of the FKBP flag. And then when we treat the cells with DTAG for 2 hours, and in fact, even for 1 hour, the protein is completely gone, as you can see, both with an anti-CBP alpha antibody as well as with an anti-flag antibody. We confirmed that this tag protein does indeed bind to this site that we had identified, and this binding is lost when we treat the cells with DTAG. And just to make sure that this peak is not some kind of bioinformatic artifact, we also did old-fashioned chip qPCR, and we can again find that there's a strong signal for CBP alpha binding at this binding site and not to a control site. And when we treat it with DTAG, this peak goes away. Now, this particular region is actually within the transcribed region of rDNA. So it would be within that Christmas tree region that I'd shown you in the EM image. And so we reasoned that there must be fragments that would be occupied both by polymerase 1 and by CBP alpha at the same time. So we did sequential chip qPCR where we first pulled out with flag, eluted the fragments, then pulled out with polymerase 1, and we could identify this exact binding site. But when we replace either of these antibodies with IgG, that binding goes away. And so CBP alpha is actually binding to actively transcribed rDNA repeats. Now, once we have this deGrund system set up, we wanted to look at the consequences of CBP alpha degradation on rRNA transcription. And there are several assays out there. I can talk about this more in the Q&A, but we optimized this assay called FishFlow, where we exploited the fact that the RNA gene is first transcribed into this 13 kilobase 47S RNA, whose tips are cleaved off to make a 45S RNA. And so we designed probes against the extreme 5' end, and these probes give a very nice, exclusively nucleolar signal that surrounds all one foci. So you can think of each of these dots as a collection of rDNA genes that are being transcribed, and this 47S is being radiated out from it. And now we can pass these cells through a flow cytometer. We can actually quantify nascent rRNA on a per cell basis. And using DAPI, we can actually gate on cell cycle and quantify nascent rRNA in different phases of cell cycle, which is really important because you actually have cell cycle phase differences. And what we found is that if we take our parental line and treat it with DTAG, there's no effect on nascent RNA, showing that DTAG is not toxic to the cells. But when we take our deGrund line and treat it with DTAG, there is a rapid reduction in rRNA transcription that plateaus at about a 1/3 reduction in the ability of these cells to make ribosomes. And if you think about the fact that each cell requires to double its ribosome content in order to divide, a third reduction is a 35% reduction is a substantial impairment in the cell's ability to make new ribosomes. And because we could see an effect even very early after treatment with DTAG, here is another sort of a closer view, and this is now a zoomed in y-axis. But I hope you can appreciate that pretty much as soon as CBP alpha gets degraded, the amount of nascent RNA in the cell begins to dip. And because we have the ability to gate on different phases of the cell cycle, we can actually analyze the cells separately. And again, you can see no matter where they are in cell cycle, the amount of nascent rRNA reduces in these cells. This leads over time to reduction in size of nucleoli, reduction in abundance of mature ribosome subunits, so this is 40S and 60S subunits, and reduction in growth of the cells. So the next question we asked is, what step of rRNA transcription is being affected by the lack of CBP alpha? And the core machinery of rDNA transcription is actually quite different from Pol1 machinery. So the first thing that happens is a subset of the hundreds of copies in the cell are occupied by this protein named UBTF, which is an HMG box protein that actually kicks out the histones and is thought to create a kind of specialized chromatin on rDNA. Then you get occupancy of a tata-binding pentameric SL1 complex, and this recruits polymerase 1 complexed with its initiation factor RRN3. And then RRN3 detaches and Pol1 migrates along the length, and you can imagine it sort of going all beyond the screen and producing those Miller spreads that I'd shown you before. So the first question we asked is the whole cell abundance of any of these factors changed? And we found that there was no difference in the whole cell abundance of any of these factors after we degrade CBP alpha. So we next look at ChIP-seq occupancy, and we first started with Pol1. And what you're looking at here is 0, 4, 8, and 10 hours, showing the average along with the standard of mean at different time points. And there was a very rapid, within 4 hours, 40% reduction in the amount of Pol1 occupancy across the rDNA gene. And you can appreciate this here perhaps a little better with bar graph showing binned occupancy across the gene. And so Pol1 occupancy is reduced with CBP alpha degradation. We then ask what happens to RRN3. So RRN3 detaches from Pol1 as it starts transcribing, and so it's really only at the promoters. And as you can see, there is about a 40% reduction here as well, matching the reduction in Pol1. On the other hand, SL1 occupancy is not changed, and UBTF, which I haven't shown you, is also not changed after we degrade CBP alpha. And what we believe is happening is this step is being impaired by the loss of CBP alpha. And so the overall model that we have is that every single cell in your body has hundreds of rDNA copies. And each one of these rDNA copies contains motif sequences for CBP alpha, and in fact, for many other factors, which I didn't have a chance to show you. But most cell types will not have a protein that is capable of binding to these cells, but to these motifs. But in certain cell types like myeloid progenitors, there's a factor like CBP alpha, which binds to this motif. And through mechanisms we really don't yet understand, it boosts the occupancy of Pol1 and RRN3 and the transcription of rRNA. Important to note that CBP alpha is not part of the core machinery of Pol1. And so it is not essential for rRNA transcription in all cells, but it may be required to boost rRNA transcription in certain cell types. Now, this, of course, there are lots of studies that we're doing to take this work forward, but just to give you a taste of some key questions that we have. The first is that right now we are degrading CBP alpha in the entire cell. We really want to be able to test that this regulation is direct. And so what we would love to do is mutate the CBP alpha motif. Now, keep in mind, for every normal gene, you just have to mutate 2 copies per cell. In this case, we have to mutate hundreds of copies. So right now we're using base editing to try to simultaneously mutate a large number of copies to test this direct regulation. We also want to do this kind of background studies in the live mouse to ensure that indeed this rRNA and ribosome biogenesis regulation is happening in vivo, and also look at what kinds of co-regulators CBP alpha is using to regulate POL1, because CBP alpha is known to recruit P300 and mediator, which may be relevant for POL2. We don't know if those complexes are also relevant to POL1. And what I wanted to end with is just mention some resources that we have put out that might be of interest to the broader community. So one of the reasons why rDNA, quote unquote, epigenetics really doesn't exist as a field is because rDNA, because no one is typically mapping the tracks to rDNA, because standard genomes do not contain intact rDNA sequences. So what we have done is taken some of the most commonly used human and mouse assemblies. We have masked rDNA fragments in those genomes, and then added a single rDNA copy as an extra chromosome R. And we have made these genomes publicly available on GitHub, and we also have a bioinformatic methods paper on how to map to rDNA. So pretty much anyone can now map to rDNA without requiring any specific expertise. The other assay which I'd mentioned was fish flow for the quantification of nascent and mature rRNA. And so we have sets of probes for human and mouse, 47S, 18S, and 28S rRNA. And we have a paper in STAR protocols that should be out soon, which should make it really easy to quantify rRNA for anyone who might be interested. So with that, I will acknowledge the members of my lab, especially Charles and Subin, who have done all this work, as well as our funding sources and our collaborators. And thanks again to the Fragile Nucleosome community.
Great, Vikram, and thank you for the wonderful presentation. So if you have any questions, feel free to type them in the Q&A box. So we got one question from Jason, and hi, really interesting talk. I was wondering so he has 2 questions. I'm going to read one by one. So the first question is whether CBPA alpha binding site is considered an enhancer, and if it loops to the promoter of 47S, and whether you think the CBP alpha degradation in itself disrupt the integrity of the nucleolus.
Yeah, so those are both great questions. So to the first question, the CBP alpha binding site is 5 kilobases downstream of the promoter. And historically, at least with high C, 5 kilobases is nothing. If you cannot resolve that amount of separation, micro C may be able to resolve 5 kilobase difference. So we are going to be looking at that. One interesting feature of rDNA, and this has now come up with super resolution microscopy images, is that each rDNA copy seems to form a sort of circle where the start of its transcription and end of its transcription forms a loop. And it is possible that areas within the loop are contacting each other. And so what could be happening is that all areas within the loop are contacting each other, and the CBP alpha binding site is also potentially contacting the promoter of the 47S. I don't know if CBP alpha is disrupting the integrity of the nucleolus. We do know that when you reduce rRNA transcription, the size of the nucleolus shrinks because the nucleolus size is proportional to the number of ribosomes being made. So I don't think CBP alpha degradation in itself disrupts the integrity, but the reduced rRNA transcription would reduce the size of the nucleolus.
Nice, and I think Ben also has a question that he's going to ask himself.
Great talk, Vikram. I really enjoyed that. The rDNA is something that I know not much about, but would love to know more about. One of the things I do know is, like, when people do experiments to look at, like, of all the repeats, how many are being transcribed, there's always like a fraction that's open chromatin and transcribed, a fraction that's repressed. And I was wondering if you've looked for CBPA, is it upregulating transcription of the ones that, like, more transcription for the ones that are open versus opening some that are closed?
A great question. So actually the UBTF occupancy is a good surrogate for the ones that are open. And so the way you can think of it as, if it's inactive, is just histone chromatinized with basically wrapped around nucleosomes. Then you have a subset that are occupied by UBTF and histones get removed. And that would be the UBTF chip-seq signal, which we find does not change when we degrade CBP alpha. So we think that the number of active copies are not changing. What is happening is the same number of copies are being transcribed at a lower level. And so, so you can imagine if we were to make Miller spreads, actually, I would love to make Miller spreads. No one is doing this anymore, apparently. But if we could make Miller spreads, what I think we would see is the same number of Miller spreads, but per cell, but each Miller spread would be more sparse because the Pol ones are entering at a slower. There are more delays between each new Pol one entering the rDNA copy.
Awesome, thank you.
So we have another Q in the box. Great talk, thank you. And I was wondering whether Pol1 levels go down as well, or that it just occupies lessly DNA.
Good question. So that's obviously the first thing we looked at, and this is Pol1. So Pol1 is a 13 subunit complex. We have only looked at its core catalytic subunit, and we do not see any difference in the amount of this subunit. Of course, we haven't looked at all 13 proteins. Maybe one of the things we should do is just do mass spec at these points, global mass spec, to see if we see any evidence. But we don't think Pol1 is being reduced, as the amount of Pol1 on the cell is not reducing. The amount of Pol1 loaded on rDNA is reduced.
So you mentioned like the CBP alpha, it's called myeloid lineage specific, and you have this motif it occupies too. Then how about the lineages that don't have it? Is it just reduced, like producing less DNA, or it's like in different lineage, it's regulated by different lineage specific TFs?
Right, and so this is, I think we are, this is really early days of this model. And so I don't know if I can give you a definite answer to this question, but the overall way I envision this going is that, you know, every cell type has a cohort of somewhere between 5 and 10 essential transcription factors that set the identity of that cell type. And I think a subset of those factors bind to rDNA and regulate rRNA transcription in that cell. And in every different cell type, different factors probably are creating a set point where rRNA is being transcribed in that particular cell. Now, each and every case is going to have to be individually tested. But one perhaps corollary of this is, you know, there are many situations where you knock out a transcription factor and you get the loss of a lineage. Typically, it's attributed to, we think that, okay, the transcriptional network in that cell is collapsing, or lineage identity genes are not being turned on. One possibility to consider is maybe that particular lineage is no longer able to make ribosomes, adequate number of ribosomes, and that is partly responsible for the loss of that lineage or some of the phenotypes. So if our model is true, then I think the rate of transcription of the most abundant RNA in the cell should actually be considered as part of the transcriptome. And anytime you're analyzing the consequences of modification of transcription factors or epigenetic regulators, we should at least ask what is happening to RNA transcription.
Then do you think if you can design like guide RNA, like use CRISPR system, then that would bypass the need of using these TFs, and then like you can rescue to some degree using CRISPR system?
Good. So that's a really interesting question. So, you know, the CRISPR-A recruits Pol2 co-activators, as far as I know. We don't know if there's a CRISPR-A for Pol, and no one has designed a CRISPR-A for Pol1. But I'm sure that, you know, we just have to find out what is the right kind of thing to fuse to dCas9 to recruit. Maybe we need to like recruit RRN3 or something like that. You can imagine that is an approach. One of the things we are trying to do is we're trying to degrade CBP alpha in the entire cell and then use a dCas9 CBP alpha fusion to only put it back on rDNA. And what that would do is it would specifically rescue it over there. So that would be another way to uncouple non-specific effects coming from global changes in the cell from specific rDNA effects. Hopefully, if you invite me back the next time, I'll be able to tell you these results.
Yeah, that's really cool. Yeah. Oh, there's one more question. Great talk from Marta. And do you see evidence of cell type regulation for specific RNA variants? And by RNA variants, I mean RNA copies that might have slightly different sequence and maybe different functions?
Yeah, so this is this very growing and rapidly evolving field. So the hundreds of copies that we have within a cell are don't have the same sequence. So there are SNPs in rRNAs, and there are SNPs between the copies within the cell. There are SNPs between individuals. There are different SNPs that are sort of in different evolutionary separated groups of humans. For example, you would see different SNPs. To what extent, and what this leads to is this leads to actually production of ribosomes with slightly different RNA sequences. To what extent these things are being regulated is simply not known. The one thing I can tell you is we looked at our CBP alpha and several other transcription factor motifs, and those sites did not have any variants that we could identify. So even though there are variants identified in human rDNA, we could not identify any variants in our transcription factor motifs. So that's all I can say. I don't think CBP alpha is activating some repeats at the expense of others because it cannot bind to them.
Cool, great, thank you. Thank you all the speakers for the wonderful presentation, and I think we are right on time. Yeah, thank you everyone for joining the mini symposium, and especially the speakers.